This is the teacher tip video for decoding DNA, modeling protein synthesis. First, a bit of background information. DNA carries genetic instructions for an organism. It's a polymer made up of adenine, thymine, cytosine, and guanine nucleotides. It is arranged in long chains that form a double helix, and on each side of the helix is a complementary nucleotide. A pairs with T, and C pairs with G. It is important to remember that DNA cannot leave the nucleus of the cell. RNA polymerase makes a copy of the DNA, producing a molecule of messenger RNA. This process is called transcription. Notice that the mRNA strand is very similar to the original DNA strand, except for U's have replaced the T's. Messenger RNA is a working copy of DNA that carries the message, but it is not important if it gets lost, as the DNA is still in the cell and more messenger RNA can be produced. Messenger RNA is read 3 letters at a time. These are called codons. AUG is the universal start codon. After the start codon, each 3-letter sequence is another codon, and each codon codes for one amino acid in the protein that will be formed. Transfer or tRNA molecules are used to read the messenger RNA. On one end of a tRNA molecule is an anticodon that will complement the mRNA codon, and on the other end it carries an amino acid that will be placed in the protein. The process of decoding the mRNA to produce a protein is called translation. This is carried out by a ribosome. Each ribosome can hold 2 tRNA molecules at a time. When they're held together, there's a bond formed between the 2 amino acids called a peptide bond. The ribosome is made up of 2 subunits. Translation begins with the process called initiation. tRNA molecule with the UAC anticodon matches the AUG. At this point, the ribosome will form around this tRNA molecule. There is now an open site in the ribosome for another tRNA molecule that matches the next codon. Here the codon CAU is complemented by the anticodon GUA. That tRNA molecule carries the amino acid histidine. A peptide bond is formed between methionine and histidine, and the ribosome slides down one codon. This allows the next tRNA molecule to attach. This process repeats itself as the protein is elongated. There are 3 tRNA molecules that do not have amino acids on the opposite end. Instead, they have a releasing factor, which releases the protein and the ribosome from the mRNA. The 3 codons that code for these tRNA molecules are called stop codons. The resulting chain of amino acids is called a polypeptide or a protein. This chain will fold to produce a 3-dimensional shape, which will determine its function. Now we will discuss the setup for this activity. First, print a complete set of tRNA cards for each group. It is helpful to use a different color for each page. The anticodons on each page all start with the same letter. In this case, all those starting with U would be yellow, G's blue, A's pink, and C's green. You can have the students cut these out, or if you have time and a paper cutter, you can quickly cut them out yourself. Arrange your room so that groups of 4 will have a place to work around a central location. Designate a table in the center of the room the nucleus. This will be where the DNA strips are located. Remind students that the DNA strips must stay on that central table as they cannot leave the nucleus. Their group's working area will be the model of the ribosome. One student from each group will move to the nucleus and perform the role of RNA polymerase and will transcribe the DNA into mRNA. This is one example of how you could set up your room. Notice there are 4 participants in each group. The lab write-up has 4 sentences to be transcribed, and this would give each participant a chance to transcribe 1 sentence. Each group will need a complete set of tRNA cards, a blank strip of mRNA for each sentence they will transcribe, typically 4. A student recording sheet. You can provide one for each student if you would like. They will also need a blank strip to record their engineered DNA that will be returned to the nucleus. To begin the activity, one student from each group will take a blank strip of paper and copy a DNA sentence into mRNA, modeling the process of transcription. Leaving the DNA in the nucleus, they will return to the group with their mRNA. In the high school version of the sentences, they will have to scan for the universal start codon, AUG, and mark the codons after that. The middle school sentences start with AUG and end with the stop codon. Once they have found their start codon, they will need to find the tRNA card that contains the anticodon that complements AUG. A common mistake is to look for a tRNA card that has AUG on it, but it needs to be the complement of AUG. On their student record sheet, make sure the students record the number of the sentence that they are decoding. Then they need to locate the tRNA card that complements the next codon. The start codon does not code for the word start. It just indicates the beginning of the sentence, and the next word needs to begin with an uppercase letter. Students should continue this process of matching anticodons with codons until they reach a stop codon. At this point, another member of their group will go find a new sentence to transcribe and bring back for the group to translate. Continue this until all 4 group members have transcribed a sentence. At this point, the students can have the teacher check their record sheet for accuracy. If the group has successfully completed their 4 sentences, the teacher can then give them a sentence to reverse engineer into DNA that will be placed in the nucleus. Being careful not to forget start and stop codons, students should arrange tRNA cards to complete the sentence. Students will then need to work backwards to determine the mRNA strand that would have coded for this order of tRNA molecules. Students can then perform reverse transcription to produce the DNA molecule that would code for the mRNA molecule needed for their sentence. When they think they have it finished correctly, they can have the teacher check their DNA strand for accuracy. If it is correct, they can place their DNA strand in the nucleus.